Welcome to episode 5 of Real Talk on the Radiant Unsanity YouTube channel, where we continue to investigate the cause of all diseases, Part 2, You Cannot Catch Germs, Bacteria, or Viruses. Based on the work of B. Wilder, written in 2006, this presentation explains the cause of disease, including Candida yeast overgrowth and cancer. The fact is, you do not and cannot catch germs, bacteria, or viruses, nor can you catch Candida overgrowth or cancer. The following text disproves the germ theory of disease, as promoted by the medical industry today, which was initiated by Louis Pasteur in the early 1800s. One day, the name of Royal Raymond Rife may ascend to its rightful place as the giant of modern medical science. Until that time, his fabulous technology remains available only to the people who have the interest to seek it out. While perfectly legal for veterinarians to use anything to save the lives of animals, Rife's brilliant frequency therapy remains taboo to orthodox mainstream medicine because of the continuing threat it poses to the international pharmaceutical medical monopoly that controls the lives and deaths of the vast majority of the people on this planet. Rife's inventions include a heterodyne ultraviolet microscope, a microdissector, and a micromanipulator. When you thoroughly understand Rife's achievements, you may well decide that he has the most gifted, versatile scientific mind in human history. The result of using a resonant wavelength is that microorganisms which are invisible in white light suddenly become visible in a brilliant flash of light when they are exposed to the color frequency that resonates with their own distinct spectroscopic signature. Rife was thus able to see these otherwise invisible organisms and watch them actively invading tissue cultures. Rife's discovery enabled him to view organisms that no one else could see with ordinary microscopes. By 1920, Rife had finished building the world's first virus microscope. By 1933, he had perfected that technology and had constructed the incredibly complex universal microscope, which had nearly 6,000 different parts and was capable of magnifying objects 60,000 times their normal size. With this incredible microscope, Rife became the first human being to actually see a live virus. And until quite recently, the universal microscope was the only one which was able to view live viruses. Modern electron microscopes instantly kill everything beneath them, viewing only the mummified remains and debris. What the Rife microscope can see is the bustling activity of living viruses as they change form to accommodate changes in their environment, replicate rapidly in response to carcinogens, and transform normal cells into tumor cells. But how was Rife able to accomplish this in an age when electronics and medicine were still evolving? Here are a few technical details to placate the skeptics. Rife painstakingly identified the individual spectroscopic signature of each microbe using a slit spectroscope attachment. Then he slowly rotated block quartz prisms to focus light on a single wavelength upon the microorganism he was examining. This wavelength was selected because it resonated with the spectroscopic signature frequency of the microbe based on the now established fact that every molecule oscillates at its own distinct frequency. The atoms that come together to form a molecule are held together in that molecular configuration with a covalent energy bond which both emits and absorbs its own specific electromagnetic frequency. No two species of molecule have the same electromagnetic oscillations or energetic signature. Resonance amplifies light in the same way two ocean waves intensify each other when they merge together. More than 75% of the organisms Rife could see with his universal microscope are only visible with ultraviolet light. But ultraviolet light is outside the range of human vision. It is invisible to us. Rife's brilliance allowed him to overcome this limitation by heterodyning a technique which became popular in early radio broadcasting. He illuminated the microbe, usually a virus or bacteria, with two different wavelengths of the same ultraviolet light frequency which resonated with the spectral signature of the microbe. These two wavelengths produced interference where they merged. This interference was, in effect, a third, longer wave which fell into the visible portion of the electromagnetic spectrum. This was how Rife made invisible microbes visible without killing them, a feat which today's electron microscopes cannot duplicate. By this time, Rife was so far ahead of his colleagues of the 1930s that they could not comprehend what he was doing without actually traveling to San Diego to Rife's laboratory to look through his virus microscope for themselves, and many did exactly that. One was Virginia Livingston. She eventually moved from New Jersey to Rife's Point Loma, San Diego neighborhood and became a frequent visitor to his lab. Virginia Livingston is now often given the credit for identifying the organism which causes human cancer, beginning with research papers she began publishing in 1948. In reality, Royal Rife had identified the human cancer virus first, in 1920. Rife then made over 20,000 unsuccessful attempts to transform normal cells into tumor cells. He finally succeeded when he irradiated the cancer virus, passed it through a cell-catching ultrafine porcelain filter, and injected it into lab animals. Not content to prove this virus would cause one tumor, Rife then created 400 tumors in succession from the same culture. He documented everything with film, photographs, and meticulous records. He named the cancer virus Cryptocytes primordialis. Virginia Livingston in her papers renamed it Progenitor Cryptocytes. Royal Rife was never even mentioned in her papers. In fact, Rife seldom got credit for his monumental discoveries. He was a quiet, unassuming scientist, dedicated to expanding his discoveries rather than to ambition, fame, and glory. His distaste for medical politics, which he could afford to ignore thanks to generous trusts set up by private benefactors, left him at a disadvantage later when powerful forces attacked him. Coupled with the influence of the pharmaceutical industry in purging his papers from medical journals, it is hardly surprising that few have heard of Rife today. Nevertheless, many scientists and doctors have since confirmed Rife's discovery of the cancer virus and its pleomorphic nature, using dark field techniques, the Nason's microscope, and laboratory experiments. Rife also worked with top scientists and doctors of his day who also confirmed or endorsed various areas of his work. By increasing the intensity of a frequency which resonated naturally with these microbes, Rife increased their natural oscillations until they distorted and disintegrated from structural stresses. Rife called this frequency the mortal oscillatory rate, or MOR, and it did no harm whatsoever to the surrounding tissues. Today's Rife instruments use harmonics of the frequencies shown on the display screen. The wavelength of the actual frequency shown is too long to do the job. This principle can be illustrated by using an intense musical note to shatter a wine glass. The molecules of the glass are already oscillating at some harmonic of that musical note. They are in resonance with it. Because everything else has a different resonance frequency, nothing but the glass is destroyed. There are literally hundreds of trillions of different resonant frequencies, and every species and molecule has its very own. It took Rife many years, working 48 hours at a time, until he discovered the frequencies which specifically destroyed herpes, polio, spinal meningitis, tetanus, influenza, and an immense number of other dangerous disease organisms. By 1934, the University of Southern California appointed a special medical research committee to bring terminal cancer patients from Pasadena County Hospital to Rife's San Diego laboratory and clinic for treatment. The team included doctors and pathologists assigned to examine the patients, if still alive, in 90 days. After the 90 days of treatment, the committee concluded that 86.5% of the patients had been completely cured. The treatment was then adjusted, and the remaining 13.5% of the patients also responded within the next 4 weeks. The total recovery rate using Rife's technology was 100%. On November 20th, 1931, 44 of the nation's most respected medical authorities honored Royal Rife with a banquet billed as the end to all diseases at the Pasadena estate of Dr. Milbank Johnson. But by 1939, almost all of these distinguished doctors and scientists were denying that they had ever met Rife. What happened to make so many brilliant men have complete memory lapses? It seems that news of Rife's miracles with terminal patients had reached other ears. Remember our hypothetical question at the beginning of this report. What would happen if you discovered a cure for everything? You are now about to find out. At first, a token attempt was made to buy out Rife. Morris Fishbane, who had acquired the entire stock of the American Medical Association by 1934, sent an attorney to Rife with an offer you can't refuse. Rife refused. We may never know the exact terms of this offer, but we do know the terms of the offer Fishbane made to Harry Hoxsey for control of his herbal cancer remedy. Fishbane's associates would receive all profits for 9 years, and Hoxsey would receive nothing. Then, if they were satisfied that it worked, Hoxsey would begin to receive 10% of the profits. Hoxsey decided that he would rather continue to make all the profits himself. When Hoxsey turned Fishbane down, Fishbane used his immensely powerful political connections to have Hoxsey arrested 125 times in a period of 16 months. The charges based on practice without a license were always thrown out of court, but the harassment drove Hoxsey insane. But Fishbane must have realized that this strategy would backfire with Rife. First, Rife could not be arrested like Hoxsey for practicing without a license. A trial on trumped-up charges would mean that testimony supporting Rife would be introduced by prominent medical authorities working with Rife. And the defense would undoubtedly take the opportunity to introduce evidence such as the 1934 medical study done at USC. The last thing in the world that the pharmaceutical industry wanted was a public trial about a painless therapy that cured 100% of the terminal cancer patients and cost nothing to use but a little electricity. It might give people the idea that they didn't need drugs. And finally, Rife had spent decades accumulating meticulous evidence of his work, including film and stop-motion photographs. No, different tactics were needed. The first incident was the gradual pilfering of components, photographs, film, and written records from Rife's lab. The culprit was never caught. Then, while Rife struggled to reproduce his missing data, in a day when photocopies and computers were not available, someone vandalized his precious virus microscopes. Pieces of the 5,682-piece universal microscope were stolen. Earlier, an arson fire had destroyed the multi-million-dollar Burnett lab in New Jersey, just as the scientists there were preparing to announce confirmation of Rife's work. But the final blow came later, when police illegally confiscated the remainder of Rife's 50 years of research. Then in 1939, agents of a family which controlled the drug industry assisted Philip Hoyland in a frivolous lawsuit against his own partners in the Beam Ray Corporation. This was the only company manufacturing Rife's frequency instruments. Hoyland lost, but his assisted legal assault had the desired effect. The company was bankrupted by legal expenses, and during the Great Depression, this meant that commercial production of Rife's frequency instruments ceased completely. And remember what a universal cure meant to hospitals and research foundations. Doctors who tried to defend Rife lost their foundation grants and hospital privileges. On the other hand, big money was spent ensuring that doctors who had seen Rife's therapy would forget what they saw. Almost no price was too much to suppress it. Remember that today, treatment of a single cancer patient averages over $300,000. It's big business. Thus, Arthur Kendall, the director of the Northwestern School of Medicine, who worked with Rife on the cancer virus, accepted almost $250,000 to suddenly retire in Mexico. That was an exorbitant amount of money in the Depression. Dr. George Dock, another prominent figure who collaborated with Rife, was silenced with an enormous grant, along with the highest honors the American Medical Association could bestow. Between the carrots and sticks, everyone except Dr. Cush and Dr. Milbank Johnson gave up Rife's work and went back to prescribing drugs. To finish the job, the medical journals, supported almost entirely by drug company revenues and controlled by the American Medical Association, refused to publish any paper by anyone on Rife's therapy. Therefore, an entire generation of medical students graduated into practice without ever once hearing of Rife's breakthroughs in medicine. The magnitude of such an insane crime eclipses every mass murder in history. Cancer picks us off quietly, but by 1960 the casualties from this tiny virus exceeded the carnage of all the wars America ever fought. In 1989, it was estimated that 40% of us will experience cancer at some time in our lives. In Rife's lifetime, he had witnessed the progress of civilization from horse and buggy travel to jet planes. In that same time, he saw the epidemic of cancer increase from 1 in 24 Americans in 1905 to 1 in 3 in 1971 when Rife died. He also witnessed the phenomenal growth of the American Cancer Society, the Salk Foundation, and many others collecting hundreds of millions of dollars for diseases that were cured long before in his own San Diego laboratories. In one period, 176,500 cancer drugs were submitted for approval. Any that showed favorable results in only 1/6 of 1% of the cases being studied could be licensed. Some of these drugs had a mortality rate of 14 to 17%. When death came from the drug, not the cancer, the case was recorded as a complete or partial remission because the patient didn't actually die from the cancer. In reality, it was a race to see which would kill the patient first, the drug or the disease. What Rife accomplished optically in the 1930s with his universal microscope, Gadsden Nasens accomplished with a combination of optics and electronics in the 1940s in his somatoscope. Born on 16 March 1924 in Roubaix, France, Gadsden displayed a predisposition to be an inventor when at the age of 5 he built a little moving auto-like toy from a Meccano set and powered it with an alarm clock spring. Later he built a homemade motorcycle and a mini airplane. Gadsden Nasens is a Quebec biologist who many believe has made fundamental discoveries relating to cancer, AIDS, and the nature of life itself. Through the use of a unique microscope of his own invention, the somatoscope, the 70-year-old French-born Canadian has discovered a primitive biological entity, which he calls the somatid. While attending the University of Lille, Gadsden nearly had his education disrupted by the German invasion. Fortunately, Gadsden and his fellow students escaped to Nice, where they carried on their education in exile. He was awarded a diploma from the Union Nationale Scientifique Française, a quasi-official institution under whose auspices the education of the displaced students continued. He did not bother seeking an equivalency degree from the de Gaulle government when French rule was restored. At the young age of 21, frustrated by the limitations of conventional microscopes, Gadsden set out to build a superior microscope. Technical assistance was provided by German craftsmen from Wetzlar, Germany, who checked out many of Gadsden's original ideas on optics. Privately, Gadsden devised the electrical manipulation of the light source. Once the technical aspects were resolved, Gadsden had the body of his microscope constructed by Barbier Bemard, A. Turin, technical specialists and defense contractors near Paris. Nasons claimed that the somatid is found in all biological fluids he has looked at, including plant sap and human blood. In a long-lost chapter of history and science, a violent controversy took place in France between the illustrious Louis Pasteur and Antoine Béchamp, a noted professor of physics, toxicology, medicinal chemistry, and biochemistry. Béchamp's work led him to discover microzymas, which were characterized by a host of small bodies in his fermenting solutions. After years of study, Béchamp came to the conclusion that these microzymas were more basic to life than cells. Even with his crude equipment, he was able to observe that the microzymas underwent dramatic transformations during their life cycle. This caused Béchamp to champion the idea that the cause for disease lay within the body. Pasteur's germ theory held that the cause came from without, so Pasteur's outspokenness helped the germ theory win out and dominate medical philosophy for the past century. Now, 100 years later, Gaston Nasons has discovered an ultramicroscopic, subcellular, living and reproducing microscope form which he christened a somatid. This new particle could be cultured outside the bodies of the host. Nasons also observed that the particle had a pleomorphic life cycle and had a 16-stage life cycle. Only the first 3 stages of the somatid life cycle are normal. Nasons discovered that when the immune system is weakened or disrupted, the somatids go through the other 13 stages. The weakening of the immune system could be brought about by a number of reasons, such as exposure to chemical pollution, ionizing radiation, electric fields, poor nutrition, accidents, shock, depression, and many more. Incredibly, Nasons' research has resulted in the association of degenerative diseases with the development of forms in the 16-stage pathological cycle. The ability to associate the disease with specific stages has enabled Nasons to pre-diagnose conditions in advance of when they would clinically appear. This discovery puts Gaston Nasons at odds with the orthodox medical philosophy today, which has embraced Pasteur's germ theory wholeheartedly. Nasons' work is repeatable. The ability to culture somatids is a bellwether to the rewriting of microbiology. Nasons stated, "I've been able to establish a life cycle of forms in the blood that add up to no less than a brand new understanding of the basis of life." What we're talking about is an entirely new biology, one out of which has fortunately sprung practical applications of benefit to sick people, even before all of its many theoretical aspects have been sorted out. Enderlein maintains that germs occur in many forms, beginning with the protete, the same microbe found by Béchamp, Nasons, Rife, and others, but called different names by each researcher, which can change into a virus, which can change then into a bacteria, and can later change into a fungus. Any of these forms, bacterial, viral, or fungal, can and do eventually break apart and turn back into the protetes from whence they came. And so it starts all over again, life. The protete never dies. This is the nature of life. It goes on no matter what. These protetes in our blood develop or change according to the condition of the body. At some stages of their development, they are outright pathogenic and parasitic. These are our internal parasites. These protetes can go in the other direction too and turn into cells we need, i.e., live cell therapy. They can help regenerate organs. These adapted necessities are called diseases, but they are healing processes, attempts to shunt a disturbed symbiosis back to the original healthy track. As these little dots, proteits change form. They can change into organisms that are more and more detrimental to the body. They become independent and no longer live in harmony and in support of their host body. As they develop their individual form, they create their own metabolism and waste products of that metabolism, which is harmful to local body fluids, causing pain and inflammation. Finally, this local process, which develops in the body's weakest organ, affects the whole body. It is not the organisms, germs, viruses, or bacteria that make you sick. It is the waste products of the metabolism of those organisms that make you sick. In reality, it is not the bacteria themselves that produce the disease, but we believe it is the chemical constituents of these microorganisms enacting upon the unbalanced cell metabolism of the human body that in actuality produce the disease. We also believe if the metabolism of the human body is perfectly balanced or poised, it is susceptible to no disease. These disease processes, these changes in the blood, are difficult to fathom at first because they only affect functioning, but not yet the structures of the body. This happens in the most diversified organs, such as manifesting headaches, high or low blood pressure, feeling poorly, unmotivated attitude, lack of appetite, drab complexion, coated tongue, wounds in the mouth, pimples, acne, sores, hoarseness, runny nose, ear noises, diarrhea, lowered capacity for seeing and hearing, depression, weak concentration, or poor memory. Bacteria are being found in the diseased tissues of all chronic degenerative diseases. These bacteria are the result, not the cause. Louis Pasteur was wrong. These organisms are not caught from the outside. They come from within. These germs come from inside the body, from tiny dots that you can see in the blood of any living thing with any microscope. All microorganisms, all living things come from these tiny dots, and all living things turn back into these tiny dots. These tiny dots themselves never die. The cell is not the smallest living thing. These tiny dots are. These tiny dots are called proteits in German and somatitids in French. There isn't even a name for them in English. Isn't that strange? If the environment in the body becomes toxic, polluted, or doesn't have the nutrients it requires to maintain health, these tiny dots hook together into long threads and change into viruses, bacteria, and finally fungi that clean up a corpse if things get that bad. This is what the bacteria, viruses, and germs are there for. They clean up old, diseased tissues. The germs are not the problem. The conditions, environment they are in is the problem. So do you treat the problem or the result? If the germ theory of disease were true, no one could ever get over a cold or flu or any other disease caused by bugs, since they would constantly be reinfecting themselves. Also, if germ theory of disease were true, we would all have all of the bugs all of the time, since they are everywhere in our environment, and so would all other life forms on Earth. Thank you so much for listening. References will be listed in the description.